Hello, everybody, and welcome to the latest installment in Contract Pharma's webinar series. I'm Tim Wright, editor of Contract Pharma, and today we're going to learn about pharma packaging considerations for radiation processing. Our webcast today is being sponsored by Nelson Labs and Sterigenics, which are both Sotera Health companies. Nelson's a provider of microbiological and analytical lab testing services for medtech and pharma companies, while Sterigenics offers its industrial sterilization expertise for the pharma and med device industries. Today's broadcast is designed to build your knowledge of pharma packaging. It'll address why the type and method of packaging is an important consideration for radiation processing, and how product packaging and presentation can affect routine processing. In addition, we're also going to learn about mass extraction, which is a relatively new container closure integrity test. And first to speak is going to be John Schlecht. He's the Vice President of Radiation Technical Services at Sterigenics. He has over 30 years of experience in irradiator operations, radiation safety, IQ/OQ validation of gamma and electron beam irradiators, radiation dosimetry, and process qualification. In his role, John has the global corporate responsibility for all technical areas of radiation. He received his BS in physics from Rensselaer Polytechnic Institute and serves on the ASTM subcommittee E61 dosimetry for radiation processing. And then after John, we're going to hear from Jennifer Giggy. She's a Level 3 validation specialist and technical consulting manager at Nelson Labs with over 25 years of medical device laboratory experience. She's worked in the microbiology, bioburden, IDs, and packaging sections at Nelson Labs and has experience with various microbial tests, including microbial limits, plate counts, biological indicator population verifications, organism ID tests, and bioburden tests. She spent the last 15 years working in the packaging group, and as one of the original packaging group members at Nelson, was heavily involved in validating all the packaging test methods and equipment and writing the test procedures for packaging tests performed at Nelson. She's a participating member on the ASTM FO2 Committee for Packaging and is registered as a specialist microbiologist with ASM. We're going to be accepting questions throughout the talk. You can send those in using the box at the lower left of your screen. Questions will be answered after the presentation in a Q&A, and this presentation will be archived on our website, contractpharma.com, for 1 year. If you're having any technical difficulties, you can type those into the box as well, and someone from the broadcast vendor will help you out. So if you're ready, John, I'll turn it over to you.
Thank you, Tim. Good morning, everyone. As Tim indicated, today we are going to discuss packaging considerations for radiation processing, plus a discussion on the new container closure integrity test, mass extraction. At Sotera Health, within our pharma offerings, we embrace packaging solutions across our businesses. Our goal is to develop and maintain effective laboratory testing and sterilization processes for your products, bringing them to market in the fastest, most efficient, and safest way possible. This slide provides a general overview of our services. Today, based on the interest expressed by our customers, this webinar will focus on 2 pharma packaging-related topics. I will cover an overview of packaging considerations for radiation processing, and Jennifer Giggy will then follow me and present part 2 that will focus on mass extraction, the new container closure integrity testing. The agenda for my part of the webinar will include a quick overview of ionizing radiation. Then we'll go over some considerations for primary sterilization packaging. We'll discuss some specifics for packaging in each of the radiation modalities, gamma, X-ray, and E-beam, including primary and secondary packaging. And we'll have a quick wrap-up with key takeaways. Also, as Tim mentioned, please feel free to submit questions at any time during this webinar. The first item on our agenda is a review of ionizing radiation. Some vocabulary terms we will use throughout this webinar. Radiation is the emission of energy as electromagnetic waves or as moving subatomic particles. Ionizing radiation is any form of radiation with sufficient energy to remove orbital electrons from atoms or molecules. There are various types of radiation, but we will only discuss electromagnetic and particle radiation in the form of gamma, X-ray, and electron beam radiation. Ionization. Ionization is the removal of electrons from atoms. This creates ions and free radicals. Ionization can break chemical bonds and can change material properties. In the picture on this slide, you can see an incident photon. In this case, it is depicted as a gamma ray. The incident radiation can be gamma rays, typically from cobalt-60, which have an energy of 1.1 or 1.3 million electron volts. It could be an incident X-ray, which are produced over a spectrum of energies, typically with a maximum energy of 7 million electron volts. Or it could be an incident electron beam, typically with an energy of 5 to 10 million electron volts. The incident radiation ejects an electron from the outer orbital of the atom. Once the electron from the atom is ejected, an ion pair is created, meaning we have a negatively charged electron that is now free from the atom, and the atom itself now carries a net positive charge because it has lost an electron. The ionization potentials for most atoms are typically less than 10 electron volts for outer orbital electrons. The energies of the incident photons or electrons, as I already mentioned, are in the sterilization process are millions of electron volts. Therefore, a single photon or incident electron can create thousands and thousands of ionization events in the material being irradiated. So, how do we harness this energy to sterilize medical devices and pharmaceuticals? Essentially, when we sterilize with radiation, we use the energy to damage DNA in living cells, and we are talking primarily about microorganisms. Radiation damage to the cell can be caused by direct or indirect action of the radiation on the DNA. The picture on the left side of the slide depicts an incident gamma ray interacting with the DNA molecule. In the direct action process, the radiation actually hits the DNA molecule directly, or the DNA molecule is hit by an electron previously ejected by the radiation. This disrupts the molecular structure, which leads to cell damage or cell death. There is also an indirect way that radiation can damage DNA, where the radiation interacts with the water molecules, which are the major constituents of the cell, releasing free radicals. These free radicals are very reactive, and when the free radicals recombine, we can create hydrogen peroxide, for example, which is toxic to the DNA molecule. The result of both direct and indirect actions is the impairment of cell function or death of the cell, and this is the process of radiation sterilization. Now that we have discussed ionizing radiation and what it is, what are some of the considerations for primary radiation sterilization packaging? How can radiation damage materials? Just as we discussed on the previous slide how radiation can damage living cells, radiation can also damage materials. On this slide, you can see an incident photon, in this case a gamma photon from cobalt-60. But it could just as well be an incident X-ray photon or electron from an electron beam. The incident radiation interacts with the material, in this example a polymer. When the incident radiation causes ionizations in the polymer, it can rupture chemical bonds. This process leads to chain scissioning and cross-linking in the polymer, which create changes to the material properties in the polymer. In some cases, these changes may be beneficial, and in other cases, detrimental change. But the point is that the radiation can change the material properties of the polymer. This slide depicts the relative radiation stability of medical polymer families. This chart comes directly from AMY TIR 17. As you look at the chart, the longer the green bar for a particular polymer, the more stable it is with respect to the radiation process. And you can see at the top of the chart, there are polymers like polystyrene and polyester, which have very long green bars, up to perhaps 100 kilograys. The kilogray is the unit we use to measure the absorbed energy in material, with 1 kilogray being equivalent to 1,000 joules per kilogram of material. Typically, in radiation sterilization, the minimum dose required for sterilization is on the order of 8 to 25 kilograys. And there would also be a maximum dose that would be imparted to the material, perhaps double the minimum dose, perhaps up to 45 or 50 kilograys typically. You can see I put a gray box around the range of typical sterilization doses from about 10 to 45 kilograms. The materials that are green across that entire range tend to be very easy to use in radiation sterilization and are good candidates for primary sterilization packaging. Things like polyurethanes, polystyrene, polyesters are very resistant to radiation, whereas polypropylene and PTFE, polytetrafluoroethylene, may have issues. Though there are stabilizers that can be used for things like polypropylene that can make it work quite well in radiation sterilization. The point is to be aware of the radiation impact for the material you propose to use for sterilization packaging. On this slide, I have a summary of some materials that you might use for radiation packaging. Some things that are very resistant to radiation are polyethylene, polystyrene, polycarbonate, and some non-polymeric materials like glass and metals are very resistant to radiation. An example of a good sterilization package might be the picture of the aluminized pouch I have there, for instance. I placed an asterisk there also for polycarbonate and glass, as they may discolor during the radiation process, though their mechanical properties are very stable. There are some ways to mitigate this, and I will discuss one of these later, for glass later on. Some more vulnerable materials are polypropylene, polymethyl methacrylate, and polytetrafluoroethylene, PTFE. PTFE, however, if used as a coating, tends to be okay, and there are radiation-stabilized types of polypropylene as well. Even though cellulose is listed as a vulnerable material on this slide, it is generally good for secondary packaging, meaning like your shipping boxes, what you ship the product to the sterilizer in. As I mentioned previously, typical radiation sterilization doses are from about 8 to 25 kilograms. And that's the minimum required dose, or the dose required to sterilize the product. Whenever you irradiate the product, there is a range of dose you can expect, both a minimum and a maximum dose, and this impacts your product and the packaging material used. This range of minimum and maximum dose can vary depending on a number of factors, including the type of product being irradiated, how it is packaged, the type of radiation being used for sterilization, and the design of the irradiation facility. We refer to this range of maximum to minimum dose as the dose uniformity ratio, or DUR. The DUR you can expect in a given irradiator can vary, but typically you can expect a DUR between 1.5 and 2.0 to be necessary in order to process the product. In other words, in a typical gamma irradiator with a minimum dose of 15 kilograms, you would want to establish a maximum dose of 25 or possibly 30 or 35 kilograms, depending on the design of the irradiator. The same would be true for an X-ray irradiator. For E-beam, the DUR needed for processing is more highly dependent on the actual product being irradiated, but a dose uniformity ratio of 2.0 for many products and packages is sufficient for E-beam. Whatever the modality being used, choose a packaging material that is radiation stable at a dose at least as high as your expected maximum dose, preferably as high as possible to facilitate operational flexibility in a production irradiator environment. One thing that's often overlooked in radiation processing is the validation of the maximum dose, and this applies to both the product and the primary packaging material. There is some guidance in ISO 11137-3 related to this, and there is also some guidance from the panel on gamma and electron irradiation on maximum dose validation, and I've provided a link on this slide to that guidance. Ultimately, it is the responsibility of the manufacturer to validate their maximum dose and ensure both their product and packaging can withstand the maximum dose through functionality testing. And as mentioned before, later on in this webinar, Jennifer Giggy from Nelson Laboratories will review some laboratory testing that is relevant in this area. Some other considerations for your sterilization process and sterilization packaging. Some properties in some polymers are more impacted in gamma or X-ray radiation versus E-beam, whereas other properties show no statistically significant difference between irradiation modalities. So, depending on what you are using for your packaging material, it may or may not be important which sterilization mode you are using with respect to material effects. In a prior slide, I showed an aluminized pouch, for instance, which would probably not show significant material effects in any of the radiation modalities. At the highest dose, temperature effects may be more prevalent in E-beam versus gamma or X-ray. Typical temperatures in gamma and X-ray are a maximum of 35 or perhaps 40 degrees Celsius, whereas the maximum temperature might typically reach up to 65 degrees Celsius in electron beam. So if your product or packaging is impacted by temperature, this should be considered. And in general, there is typically little difference in material effects between gamma and X-ray radiation. Next, we are going to go over the packaging considerations for the gamma sterilization process. We will focus on the secondary packaging, or the shipping containers, and how this can impact the process. In the case of gamma radiation and also X-ray radiation, we are talking about electromagnetic radiation, which is the flow of photons, or light quanta, through space. On the chart on the top of the slide, you can see the entire spectrum of electromagnetic radiation, with gamma and X-rays being high frequency and shorter wavelength radiation, and of sufficient energy to be considered ionizing radiation. Photon radiation has no mass or charge and can be produced either through natural or man-made ways. In the case of gamma radiation, it is a natural radioactive decay process we are talking about, typically from the isotope cobalt-60, that emits gamma photons of approximately 1.1 and 1.3 million electron volts. X-rays, on the other hand, are machine-produced photons. On this slide, I have a depiction of a typical gamma irradiator. On the right-hand side is a loading area for product, where the incoming shipping boxes are placed into irradiation containers. These containers are sometimes called totes, but the containers could be constructed in various ways and could even be the incoming product pallets depending on the design of the irradiator. The irradiation containers are conveyed into the irradiation cell, which is depicted on the left side of the in a cutaway view. And you can see the irradiation containers arranged in what is called the source pass. The source of radioactive material containing the cobalt-60 sources is raised out of a pool of water and into the irradiate position, typically between rows of irradiation containers. The irradiation containers will reside in each position around the source for a set amount of time, called the cycle time. The longer the cycle time, the higher the dose achieved in the product. Each irradiation container will sit in each position in the irradiation cell for the designated cycle time until it has traversed the entire path, and then it would exit the irradiator. Gamma radiation is an isotropic source of radiation, meaning photons are emitted in all directions. On the left-hand side of this slide, you can see an overhead view of an irradiator, where we are looking down at the source pass, where the irradiation container is queued up around the source of radiation, and you can see that the radiation is emitted in all directions. The two rectangles on the right side of the slide depict dimensionally what a typical irradiation container might look like. In this case, totes that are approximately 2 feet wide by 4 feet long and 3 feet in height, or approximately 25 cubic feet. Typically, as depicted, the irradiation containers would be irradiated from both sides in order to achieve a more uniform distribution of dose throughout the container. This graph shows the depth dose curve for gamma, X-ray, and E-beam irradiation modalities. The dashed red line is the depth dose curve for cobalt-60 gamma photons. On the x-axis, we have used the unit of grams per square centimeter, or what we would refer to as areal density. If you were to assume that the material we are irradiating is water with a density of 1 gram per cubic centimeter, then the x-axis can simply be viewed as centimeters. So for cobalt-60 photons, you can see that the maximum dose is at or near the surface of the material or package that we are irradiating, and then drops off to about 50% of the surface value at between 10 and 12 centimeters of water. The depth dose curve for X-ray photons, depicted by the green line, is nearly identical to that for the depth dose curve for cobalt-60. For E-beam radiation, however, the depth dose curve is quite different, as depicted by the blue line. This is because electrons are charged particles, and the way they interact with the material is quite different from a penetration standpoint, though all three modalities are quite effective when it comes to sterilization. For the case of electron radiation, you can see that the maximum dose is not at the surface, but is several centimeters inside the surface, and then rapidly drops to zero at about 5 centimeters of water. So, in gamma sterilization, how do we generate a reasonable process for your product given the depth dose curve discussed on the previous slide? As previously discussed, with respect to dose uniformity ratio, we have to be concerned with the minimum and the maximum dose achieved in the product due to material impacts to both the product and the packaging. We want to keep the dose uniformity ratio as low as possible. We typically irradiate the material from two sides in gamma processing. If we irradiate the material from a single side, this is what the dose profile on this slide is what it would look like through the irradiation container. A high dose on the surface, the right side of the graph, and lower doses as we penetrate through to the left side of the irradiation container. If we then irradiate the container from the other side, we would have the opposite dose distribution, a high dose on the left side and lower doses as we penetrate through to the right side. And of course, the result would be this green line. The result of these two overlapping depth dose curve is the green line that you see on this after we irradiate the material from two sides. And as you can see, the overall relative dose is a maximum of something like 1.1 and a minimum of something like 0.6. And if we translate this into a dose uniformity ratio, or DUR, the result is a DUR less than 2.0, whereas it would have been much higher if we had only irradiated from one side. Typically, gamma radiators can achieve dose uniformity ratios, or DURs, of 1.6 or better, depending on the design of the irradiator. What should you consider before packaging your material for the gamma irradiation process? You should be aware of the type of irradiator being used. Is it a tote or a pallet irradiator? How will your shipping boxes fit into the irradiation containers? What are the capabilities of the irradiator? What dose uniformity ratio can you expect for the bulk density of your product? Does your product represent any challenges to the process? Some examples of challenges are significant regions of high-density materials, such as large metal masses. Does the product need to be refrigerated or frozen during the irradiation process? Or does the product have a hazardous component that must be considered when handling the product? How is the product arranged in the shipping container, and will this impact the validation process? Ultimately, how will the way your product is packaged impact the process and ultimately the cost of sterilization? So next I'm going to go through some examples for the gamma process. Here are some examples of shipping boxes or containers and how they might fit within a given irradiation container. On the top of this slide, we have an example of a tote irradiation container that is approximately the same dimensions I mentioned before, 25 inches wide, 48 inches long, and 40 inches in height, or about 25 cubic feet in volume. In this example, we are irradiating a low bulk density material. It's less than 0.1 grams per cubic centimeter in bulk density, with a relatively wide dose uniformity ratio, greater than 1.6. For example, 15 kilogram minimum and 30 kilogram maximum. In this case, since the product has a wide dose specification and is low in bulk density, we can pretty much fill the irradiation container. In theory, if the boxes were the right size, we could completely fill the irradiation container. In this instance, we ended up with a fill efficiency of 78%, which is quite good. On the bottom, we have an example of a pallet-type irradiation container. And again, the bulk density is low and the required DUR is high, greater than 2.0 in this case. And we're able to achieve a fill efficiency of this one of greater than 86%. So it's almost a completely full pallet. In the next example, we have the same tote irradiation container as the first example, but a higher bulk density product of 0.4 grams per cubic centimeter and a lower specified DUR of less than 1.6. An example, 12 kilogram minimum and 17 kilogram maximum. In this example, in order to meet the specified dose uniformity ratio, the product had to be significantly center-loaded in the irradiation container in order to reduce the effective density of the product. And we were only able to achieve an irradiation container fill efficiency of 29%, or a little less than 1/3 of the irradiation container being utilized. You could see how that would impact your cost of sterilization. In the example on the right side of this slide, Even though the product had a low bulk density of just 0.08 grams per cubic centimeter, the dose specified was very tight, a DUR less than 1.4, and the product had to be center loaded, and the resultant fill efficiency due to that center loading and the size of the cartons was only 18%. So it is a combination of factors, including the bulk density of the material, the dose uniformity ratio based on the specified minimum and maximum dose, and the shipping box dimensions themselves, and how those fit in a radiation container, that can all impact the process and ultimately the cost of sterilization. And the last example is a palliative radiator again. When we think of a palliative radiator, we think, this is great. I can ship my product in the same way I'm going to process it, and there will be less handling, less chance for errors, and less chance for damage. And that may be true in some cases, but again, it is all dependent on the factors I mentioned on the prior slide. In this case, we have a pretty low bulk density product, but the specified DUR is not achievable in the palliative radiator. If you think back to the depth dose curve, a pallet is typically much wider than a typical tote, so the dose uniformity achievable in a palliative radiator is higher than in a total radiator. So we end up having to download the pallet to essentially 1/3 of the available volume in order to process the product and achieve the specified dose. There are also specialty irradiators that can irradiate materials to dose uniformity ratios much less than 1.4, even down to 1.1 dose uniformity ratios, depending on the bulk density of the material and the size of the package. But it is a very inefficient process and quite expensive, as you will essentially be irradiating a single box at a time. Next, I'm going to talk about a couple of examples of the irradiation of pharmaceutical products where the irradiation process negatively impacts the product. And in order to mitigate those negative effects, the packaging needs to be changed, either the primary packaging or the secondary packaging. In this example, we have some gel-filled syringes that are irradiated to a dose of 25 kilogram minimum and 40 kilogram maximum, or a 1.6 DUR. This is not real material. It's before irradiation, it's like mustard, and after irradiation, it's more of a clear gel you can see. So on the left, you see some mustard-colored gel before irradiation, and then on the right, after irradiation, you can see that the gel is basically clear. It's also undergone a viscosity change. So you have a color change and a viscosity change imparted to the material from the irradiation process. So how are we going to mitigate this? What are some methods we can use to mitigate these impacts to our product? One way is to maybe just lower our minimum sterilization dose to 15 kilograms. If we lower our sterilization dose to 15 kilograms, we can also lower our maximum dose because we know we can maintain a dose uniformity ratio of 1.6. So if we're able to lower our sterilization dose by revalidating our sterilization dose to 15, we can also lower our maximum dose to 25. So that may result in a better product after irradiation. We could also center load in the irradiation container. And if we center loaded and lowered our sterilization dose to 15 kilograms, we might be able to only achieve a maximum dose of 20 kilograms, or a dose uniformity ratio of 1.3. So before we were irradiating the product to something like 40 kilograms, now we're only irradiating it to 40 or 50 kilograms, now we're only achieving a maximum dose of 20. That may result in a better product. However, simply lowering the dose of the product doesn't work. We can look at irradiating the product in a frozen state, which often helps reduce the damage to many products, and that's the picture of the cryo container with the dry ice down in the lower right-hand corner of the slide. So irradiating product in a frozen state often helps reduce the damage to many products, especially gels and liquids. Of course, to irradiate in a frozen state, we may need to use cryo containers of some sort, and this will impact your secondary packaging configuration. We can also look at an inert gas overlay for the product. Inert gases such as nitrogen can reduce oxidative damage that occurs during the irradiation process. This would require a primary package that could accommodate the inert gas. In the second example for gamma irradiation, we have an active pharmaceutical ingredient in a glass vial that we irradiated to a 15 kilogram minimum and 30 kilogram maximum, or a DUR of 2.0. And we can see that before irradiation on the left-hand side, we have a clear vial, and after irradiation, the glass has turned brown. How can we mitigate the discoloration we experienced in the glass vial? Just like the prior example, we can reduce the maximum dose in the product that the product experiences during the irradiation process by reducing the sterilization dose or through packaging changes or how we present the product to the irradiator so that the maximum dose to the product is reduced. That may not be enough to reduce the discoloration in this case, and we may need to consider using a different primary package, something other than glass. However, we could also look at purchasing glass vials that are doped with cerium oxide, for instance, which will reduce the discoloration from the irradiation process. Next, we're going to discuss really briefly the packaging considerations relative to the X-ray sterilization process. If you recall the depth dose curve that I presented earlier, the depth dose profile for X-rays is very similar to that for cobalt-60. The main difference between gamma radiation and X-rays is the source. In the case of gamma radiation, the photons are emitted by the radioisotope cobalt-60, and the source is isotropic, whereas X-rays are produced by a machine and are much more focused in a single direction. For most applications, there should be little or no significant differences in gamma and X-ray and packaging or material effects. There is a caution, however, and that is if you validate your process in one modality, like gamma, you must be able to provide evidence that changing to a new modality, like X-ray, has no negative impact on your sterilization packaging or product. And lastly, we are going to talk about packaging considerations for electron beam processing. Remember, electron beams are particle radiation. Instead of electromagnetic waves, we are now talking about fast-moving, highly energetic particles, electrons with a negative charge that move through space. Particle radiation can be emitted by unstable atomic nuclei. However, in the case of electron beams, they are machine-produced in what we call an accelerator. The pictures at the bottom of this slide depict the inside of an IBA rototron accelerator on the left-hand side and a type of linear accelerator on the right-hand side. In both cases, there is a vacuum chamber wherein the particles are accelerated through some means to relativistic speeds and energies in order to be used for the electron beam irradiation process. Once again, the depth dose curve for electrons is significantly different than for the photons from cobalt-60 or the typical X-ray irradiator. As depicted by the blue line in the curve, electrons from a typical 10 MeV accelerator can only penetrate to a maximum of less than 5 centimeters of water. One of the really nice things about the electron beam process is that the irradiation container is the shipping box. As depicted by the schematic on the left, the shipping container is conveyed under a focus beam of electrons, which scan across the box as it moves under what we call the scan horn. The box may only be under the beam for 10 or 15 seconds, for example, so the process for a single box is very fast. On this slide, you can see the different depth dose curves of various energy electron beams. Typically, we use 10 MeV electrons for the radiation sterilization process, as anything lower would not be very useful for many products due to the reduced penetration at lower energies. And anything higher than 12 MeV has the potential to induce radioactivity into the product. So how do we harness this E-beam energy to get a nice uniform process? For certain optimal situations, electron beam can yield excellent dose uniformity, even approaching a DUR of 1. However, the further you get away from this optimal combination of package depth or thickness and density, the worse the DUR becomes. Just like gamma radiation, we typically get a much better dose uniformity if we irradiate from two sides. And the example above is for a product irradiated from two sides, where we can achieve a DUR of 1.36. There is a caution here, however, in that you can achieve very good DURs in E-beam, but small changes in packaging and product densities can lead to significant changes in DUR. Additionally, small areas of high-density materials, such as metals, powders, and liquids, can be challenging, and in some cases rule out E-beam as a sterilization method. And here's one example for electron beam sterilization, where we have a vial with only 1 centimeter of active pharmaceutical ingredient that we need to, it's powder, that we need to penetrate. It's only 1 centimeter thick. In this case, we can only irradiate from one side because the powder is sitting at the bottom of a vial. And if we turn the vial upside down after irradiating from one side, we would not be able to determine the dose to the product because the powder would move when we flip the boxes over. After irradiating a single vial from one side, we achieved a DUR of 3.5. This could be problematic if the product specifications required a DUR of 2.0 or even less, for instance. There would also likely be an even worse DUR achieved if we irradiated the vials grouped together in a box, like that depicted at the bottom center of the slide. There are edge effects that can impact the vials at the side of the box in E-beam, and that might require that the vials in the box outlined in red would need to be dummy or placebo product instead of the active pharmaceutical ingredient. So again, how would we mitigate these issues? With the product packaged the way it is and the radiator we are using, we can only irradiate from a single side, and we cannot achieve the dose specification in a straightforward way. We can't take advantage of the two-sided depth dose curve depicted on the right-hand side of the slide because we're stuck with the depth dose curve on the left-hand side for single-sided irradiation. One thing we might do is change the primary packaging in some way so that we could irradiate the product from both sides. We could try to validate a minimum dose that's lower or a higher maximum dose, but that might not be practical in this case. We could use an E-beam irradiator that irradiates the materials from both sides without the need for flipping the box, as there are some designs that allow for this. Or we could ultimately decide to use gamma or X-ray for this process. One other consideration for processing in E-beam and other modalities as well is whether or not your product has a hazardous component, as it is necessary in E-beam for sure to invade the product during PQ dose mapping of the process. So it might be necessary to use placebo product that has the same density, in this case, as the API, in order to do the dose mapping validation of the process. And lastly, just a quick wrap-up and some key takeaways from my part of the presentation. So I hope the key takeaways for today are that package design should be considered early in the development process of your product in order to have a successful radiation sterilization process. And packaging considerations for electron beam can be different than gamma or X-ray. Primary packaging is readily available, but must be resistant to the doses and temperatures encountered during radiation sterilization. And finally, consideration of the final packaging, both materials and configuration, early on in a project will help ensure a successful radiation sterilization process and will help you reduce project cost. And next, I'm going to turn it over to Jennifer Giggy, who will discuss the new container closure integrity testing mass extraction. And please don't forget that you can submit questions at any time during the webinar. Thank you.
Thanks, John. It's a pleasure to speak to you today about mass extraction. If you have particular interest in container closure integrity testing, then I'd highly encourage you to check out the upcoming Pharmaceutical Sterility Assurance Seminar we have. It's a 3-day seminar in September, and you can find the agenda and registration information on our website, www.nelsonlabs.com, under the News and Events and then the Seminars tab. I'll be speaking more extensively in that seminar on container closure integrity testing overall and going through all the available testing options. But today, due to time constraints, we're only going to be talking about mass extraction, which is one of 3 container closure integrity tests that we perform here at Nelson Labs. Mass extraction is a CCI test or a container closure integrity test. These are tests that are designed for rigid containers, such as vials and syringes and bottles, and CCI tests are used in place of traditional sterility type tests as part of a stability test program to show that these containers are sterile and that they'll remain sterile through the expected use life. CCI tests are divided into 2 groups. The first group is probabilistic methods. These are methods where the result is a pass or a fail, and they're based on visual observations by humans. The second group would be deterministic methods. These are methods where the result is a measured number that can be compared to a standard value. In other words, my result of X is either higher or lower than a PassFail threshold value. If you're above the number, then the result's a fail, and if you're below the number, then the result is a pass. This list here in the slide comes from USP 1207, and in the seminar in September 2020, we'll go into detail about how all of these method options work. The latest version of USP 1207 recommends deterministic methods for CCI testing, and this is really driving a shift in the industry towards deterministic methods. You can see why deterministic methods are preferred. They're giving a measurement, which not only tells you if you passed or failed the test, but how close or maybe how far that magic pass-fail threshold value you are. Deterministic methods are usually non-destructive, which allows for 100% inline testing during the manufacturing process. And this is especially useful since most typical failure rates in vial and syringe containers are really low and not likely to be detected unless large number of samples are tested. In a laboratory setting, this means that failing samples can be retested and investigated for failure cause. The smallest detectable defect size is usually smaller with deterministic methods. In contrast, the probabilistic methods are subjective. You're relying on a human to make a judgment call versus a machine to measure a value. Probabilistic methods are more prone to contamination because the container usually needs to be opened in some way during the test, and this is what makes those methods destructive. Finally, as discussed earlier, the minimum defect size is usually larger with probabilistic methods. So why would anyone choose to use a probabilistic method given the clear advantages of deterministic methods? Well, the answer is usually cost. A probabilistic method can usually be set up for maybe 5,000 to 10,000, where deterministic methods, the equipment alone is more than 100,000. Mass extraction is based on the mass conservation law. This law states that mass flow will not change in a closed system. So if you measure the makeup error that's required to hold the vacuum in a closed system, then that will equate to the leak rate. A smaller leak rate would equate to a smaller defect size. The leak rate is influenced by a number of factors, like the amount of free space in the test chamber and the test method that's used, and it's going to slightly vary from one test configuration to another. So there isn't a single flow value that equates to a certain defect size. That needs to be determined with each test setup. What is mass flow? It's actually very similar to volumetric flow, which is more commonly understood. Volumetric flow is a volume change over time, and it's usually expressed in units like cubic centimeters per minute. Mass flow is mass change over time, and that's usually expressed in units like grams per minute. For mass extraction, the flow values are expressed in micrograms per minute because the mass extraction detector is capable of measuring to that sensitivity. So to review, the output or results for mass extraction test are flow values in micrograms per minute. It's a flow value, but it's measuring mass to determine mass flow. Here's a bench version of a mass extraction instrument from ATC or Advanced Test Concepts. They're the company who pioneered the test method. Most mass extraction instruments are custom built for inline manufacturing, but ATC offers this model for bench testing. Let's look at what we need to run a test. The first thing is we need a chamber. The chamber is connected directly to the instrument and it holds the sample. We have a few different chambers for different sample types. Next, we need a master. The master is a solid metal part that's similar in size and shape to the test sample. Since it's a solid metal part, we know that it has no leaks. We use the master to verify that the test system is clean, dry, and free of virtual leaks or residual mass. If the master doesn't pass the test, then that indicates that there's something wrong with the test system that needs to be removed or corrected before the samples can be accurately tested. Finally, we need some void fill. Free space in the chamber will affect the test, so we want to minimize free space as much as possible. These void fill parts are also solid metal, so they have no leaks. We have a variety of different filler options for each chamber, so we can use a variety of sample sizes with each chamber. The CLO is a calibrated leak orifice. It's a defect of known size that's incorporated into the instrument. There's a switch on the instrument that turns the CLO on or off. When the CLO switch is off, the CLO path is closed. When the CLO switch is on, the test goes through the CLO, so the result is equivalent to having a 2 or a 5 micron defect in the sample. Having a built-in defect is really handy because it eliminates the need to create defects in the actual test samples. Intentional defects from laser drilled holes, microtubes, or sealing wires in the closure have some inherent problems. So this is a feature that we really love about this instrument. The CLO is our positive control for the test. A CLO value is generated by running the master with the CLO switch turned on. And this is done with every test set, so we have a flow value for a defect of known size in the specific test system. Test methods control how the test works. All methods follow the same series of steps. The timing for each step can be changed in the individual methods as well as the limit values. The picture here in the slide shows an example of a vial method. This vial method takes about a minute to complete. Different sample types and sizes may require adding or subtracting from each time spent in each step to create a suitable test method. For an inline manufacturing mass extraction test setup, where the fixtures are built specifically for a single sample type, the method can be optimized to run in just a few seconds. We've opted for slightly longer test methods so that the method can be used for a wider variety of test samples. How does a test method work? To begin with, the instrument draws a rapid vacuum as part of the gross leak check. This is 2.3 seconds in our vial method example. If the vacuum reaches stabilization before the time is up, then the test will progress to the next step. If the stabilization isn't reached, then the test will terminate. The next leak check is the large leak check. Both leak checks are mainly designed to protect the instrument from moisture contamination. And then this is followed by a long stabilization phase. The last part of the test is a fine leak check, which is where we're looking to detect the defects down to a 2 micron or 5 micron size. For the fine leak check, the mass flow is diverted through the IMFS, or Intelligent Molecular Flow Sensor, which is a super sensitive detector that was developed by ATC to measure mass flow. The IMFS records the final mass flow at the end of the test, and then this value is compared to the PassFail Threshold value for the method. If you're over the limit, then you fail, and if you're under the limit, then you pass. This slide has a link to a short conceptual video from Pfeiffer Vacuum, who bought ATC. Unfortunately, I can't get that link to work in this presentation, but if you go to YouTube and search for mass extraction Pfeiffer Vacuum, then this is the first video that will pop up. And you can take a look at that. Where is this pass-fail threshold coming from? There's no one number that's appropriate for all tests, so the value has to be determined through validation of the method with a certain sample type. A set of non-leaking or good parts are tested with the method, and those final flow values are determined. When the samples are run again with the CLO on to simulate a defect of known size. These two sets, the good parts and the defective parts, are then statistically compared to one another, and a value somewhere between the sets is calculated as the pass-fail threshold. Now, let's go over how a typical test would be run. First, we would select the chamber, the master, and the void fill that most closely represents the test samples from our library of accessories. Then we would pick the test method. As we talked about earlier, the methods are customized for the sample type. So if our sample was a vial, then we would pick the method that we have validated for vials. The first step in testing is to make sure that the instrument is free of residual mass. Remember that any mass in the system will look like a leak to the instrument, so we verify that that system is clean and dry by running the master. We know the master can't leak, so if the master fails the test, then we need to clean the system. Once the master passes, then the system is ready to go. Next, we run the CLO to determine a final flow value for a 2 micron or a 5 micron leak. Again, the leak size depends on the method. The vial method is validated for a 2 micron leak, so we would run the 2 micron CLO. And finally, we would run all the test samples. Here are some graph examples of typical vial test data. The flat part at the front of the curve is the gross leak check. The larger of those 2 peaks that follow is the large leak check. The long flat line is the stabilization phase, and the rise at the end is the fine leak check. The CLO value in these graph examples is about 12, and this would be a 2 micron defect. And you can see here that all the passing samples have values of 2 or less. The final report will contain some critical method parameters, including the PassFail Threshold value and the pressure limits and some timing steps. Final flow values and the corresponding pass or fail determination is given for each sample in the results table. The CLO value is also reported so you can compare it to your sample results. That wraps up this part of the webinar on mass extraction testing. I noticed we've had some questions come in, so let's see if we can get some of those answered.
Yeah, thanks a lot, Jennifer. Great presentation, and thanks, John. Yeah, we have some questions, and we're running up against the top of the hour, so I'm just going to jump right into them here. And the first one will be for you, John. What do I need to be aware of before initiating any change to product packaging? Are you there, John?
Yeah, I'm here. Can you hear me?
Yep, gotcha now. Did you hear the question?
Yes, I did. Thank you.
Okay.
All right, so a couple of things. One, you have both your primary and your secondary packaging. So you have your primary packaging as your sterile barrier. So any change to that primary packaging, obviously you need to assess it for function. You probably have to repeat functionality testing or have some evidence that the sterile barrier is still good for whatever radiation modality you're using. So you're going to have some evidence that a change to your sterile barrier didn't impact the sterility of your material. Then, of course, you have a secondary packaging. The secondary packaging, meaning the shipping boxes, what you're shipping it to the sterilizer in, whatever that is. You have shelf boxes, you have things that you have corrugate that you put the material in and put it on pallets and ship it to your sterilizer. That can impact, change to that can impact the sterilization process significantly as well. So either one of those you need to communicate to your sterilization vendor. So the change in the secondary packaging and the primary packaging need to be communicated to your vendor, as it may impact the sterilization process. And a change to your primary packaging, you're going to need to validate and ensure that it doesn't have an impact with respect to the sterility of your product through functionality testing.
Okay, great. Let's go over to Jennifer. What types of samples are suitable for the mass extraction test method?
Yeah, mass extraction can be used on a wide variety of samples. It has been used for both flexible and rigid containers. At Nelson Labs, we've got the fixtures and the methods to do vials, syringes. We do a lot of auto injectors. We're also in the process of getting a IV bag fixture, so we can start testing IV bags sometime later this year.
Okay, thank you. And let's see, back over to John here. Does the discoloration of glass have any impact on the chemical durability?
I will say with 99% assurance that no, it does not. In fact, there are certain things I know in the past we've irradiated that the color change was actually looked at as a positive in certain regards. So If you recall, I presented a slide on the radiation impacts to various materials, and glass was actually there as a very compatible with the radiation process from a materials standpoint. It's just the discoloration. So as far as I know, I can't say with 100% assurance, but as far as I know, there is no impact to the glass itself as far as the properties of the glass other than the changed color.
All right, thank you. Another one here for you, John. How does electron beam energy, either higher or lower than 10 MeV, affect depth of penetration or DUR?
Okay, I think I can change. I think I can change the slides. Correct? I can go.
Yeah,
back
you can
up.
go. You can go back.
So I think it'd be best to show this on a slide, and I'm going to go back to this slide. So this slide actually depicted 3 different energies: a 5, a 7, and a 10 million electron volt electron beam. And typically in radiation sterilization, we use 10 million electron volts or 10 MeV beam. And the reason is, as depicted by the red bar, the red line, is the penetration is better. You can see that the dose builds up, reaches a maximum somewhere around 4 centimeters of water, and then rapidly drops off to 0 at about 5. And again, this, when I say centimeters of water, if it was a different density material, this curve would be shifted one way or the other. But a way to look at it is just assume this is water. And if you go to the dashed orange line, at 5 MeV, you will see that the dose builds up to a maximum at less than 2 centimeters and then drops off to 0. So what that's telling me is if I wanted to sterilize something with 5 MeV electrons, it would be more challenging. Remember the example I gave of the 1 centimeter of active pharmaceutical ingredient? That was with 10 MeV. We still had a problem with the DUR being 3.5 in that example. Well, with 5 million electron volt E-beam, you probably would reach, or its potential, you would reach 0 or a very low dose. It would maybe not 0, but that 3.5 might go to 10 or 20. It would be very difficult to penetrate the material with a lower energy electron. And if you recall further back, I had also indicated that you don't want to go higher. About the highest you want to go with electron beam is 12 MeV, because above 12 MeV you you can actually make your material radioactive, which obviously is not something you want to do.
Okay, great, thank you. Let's see, back over to Jennifer. We have time for 1 or 2 more here. Jennifer, could you please compare the sensitivity of helium leak test and mass extraction test?
Sure. Helium leak can be run sometimes as a probabilistic method if you're using it more in the sniffer mode, or it can be a deterministic method. So if it's being run as a deterministic method type setup, then it's going to be more sensitive than if it's run as a probabilistic. So if I'm comparing the deterministic helium leak and the mass extraction, they can be very comparable. All of those deterministic methods are usually 10 micron leak or less that they can detect, but the sensitivity of the test, any deterministic test that you use, will be varied depending on how your test is set up. Mass extraction, for instance, can be as sensitive as 1 micron leak, but that depends on the chamber that you're using, the method that you're using, and all of the fixturing. So I would say if they're a deterministic, both side by side, they're probably pretty comparable for the same sample type.
All right, great. Thanks so much. So yep, we're right at the top of the hour here, so I'm going to end it here. If we didn't get a chance to get to your question, both Jennifer and John will have those and will respond to you directly. Thanks again, Jennifer and John. Great presentation, very informative. And thanks again to Sterigenics and Nelson Labs for the sponsorship. And thank you to all of you in the audience. I appreciate your attendance. And as I mentioned earlier, this session will be archived for 1 year on our website, contractpharma.com. Thanks very much again for joining, and have a great rest of your day, everybody.